did this project as a member of the Howard University Environmental Biology Scholars, which is an NSF-funded program. And to give you a little background about the project, Alyssum murale is a hyperaccumulating plant. And so that means that it grows on serpentine soils, and serpentine soils are characterized by high levels of heavy metals and very low levels of nutrients. And so these soils are home to many hyperaccumulating plants, which are able to take up those heavy metals and sequester them in their above ground tissues. And Alyssum murale in particular is a nickel hyperaccumulator that is native to the Mediterranean region. And so I wanted to look at the fungal relationships in Alyssum murale because the winters in the Mediterranean are really cool and wet, and so it leaves the plant very susceptible to different fungal interactions. And so that's why I wanted to do this project. And so I had two questions for this project, and the first was, do plants growing in soil with high nickel levels have greater resistance to fungal pathogens? And then the other question was, does mycorrhizae influence the growth of Alyssum murale seedlings across varying nickel concentration gradient? So there were two different experiments. The first one I'm going to talk about is the fungal pathogen experiment. And in this experiment, we took a pathogen that had grown in the greenhouse in like 2006. When they came back from Christmas break, the plants were completely covered and infected. And so I took that and I ground that up and I applied it to some Alyssum murale seedlings. And across varying nickel levels at a gradient. And so what we wanted to observe was, would the higher nickel levels have better defenses against the fungal pathogens? And so what we found in that particular experiment was that plants in high nickel soils, the 500 parts per million and the 2,000 parts per million, they had significantly more leaves and greater shoot weights. So the nickel did exactly that. It did actually help the plants defend against fungal pathogens. And they had higher they had higher leaves and higher shoot weights than the lower nickel levels, which was 0, 50, and 100. So those were the 5 levels that we looked at in this particular experiment. And what we wanted to do, because it's always suspected that the nickel in Alyssum murale helps defend against herbivory, but we wanted to know if it could also defend against fungal pathogens. And from this experiment, it definitely looks like it does. And I did another experiment looking at their response to mycorrhizae with Alyssum murale. And I did this experiment because I wanted to know if perhaps mycorrhizae was responsible for helping the plant take up so much nickel. So we know it takes up nickel, but we don't necessarily know how or why or what mechanism. So I was thinking that perhaps AM fungi was the reason why, and that might have been a stretch because this plant is in the Brassicaceae family and they don't often form mycorrhizal associations. But so we did the AM fungi experiment, and we did 4 nickel levels. So we looked at it at 0 parts per million, 100 parts per million, and 500 parts per million, and 2,000 parts per million. And we inoculated it with AM fungi mix with liquid, and we put it over the seedling, so we let it grow with the AM fungi, and we let them grow over 14 weeks. They were rotated daily and watered daily. And with that experiment, there was no difference seen in germination with and without AM fungi. Actually, at the 0 parts per million level, the plants actually did worse. So it looks like AM fungi was acting as a carbon sink and taking nutrients from the plant, but it never helped the plants grow any. Acknowledge the University of Virginia Blandy's Experimental Farm for helping me do this experiment over the summer that I was there. Also, my lab mates, Alexandria Igwe, Ebony Hampton, Chandler Purity, and Nia Johnson, Dominique Palmer, and our greenhouse manager, who was a huge help, Maria Ramos, as well as Dr. Mary McKenna and the National Science Foundation. Thank you.